Imagine buying a pair of shoes. Would you settle for any size off the shelf, hoping it might fit? Probably not, right? We expect our clothes, our food, and even our technology to fit our needs and preferences. So, why should healthcare be any different? In the world of medicine, a one size fits all approach only works some of the time. And if trying to treat melanoma some of the time isn't enough. The traditional methods have brought us far, but what if we could go even further? Hi, my name is Shreya Santhosh, and I'll be presenting on how we can use personalized immunotherapies to advance melanoma treatment. My mentor for this project was Connor Dunn, who I want to acknowledge for helping me so much throughout this process. I'm presenting from Sammamish, Washington, and attend Tesselson High School as a sophomore. So over the years, I've been interested in medicine, and here are some activities that help pique this interest. And melanoma specifically, I was really interested in making the treatment less draining and more successful because it has such a high mortality rate. So with this, let's move on to my research questions. So one, what are the limitations of traditional melanoma treatments? Two, which parts of our body needs to be enhanced to fight melanoma quickly? And three, how can melanoma treatments be modified to maximize efficiency? So here are some key definitions that are necessary to know. Melanoma is a skin cancer that is characterized by its rapid progression and high mortality rate, and is responsible for 75% of skin cancer-related deaths in the United States. And it originates from the uncontrolled growth of melanocytes, which are the pigment-producing cells in your skin. And this is often caused by UV exposure or genetic predispositions in the BRAF or NRAS genes. And these encode proteins in important cell signaling pathways. And immunotherapies are cancer treatments that boost the body's immune system to fight cancer cells more effectively. So, what is the difference between immunotherapy and personalized immunotherapy? Traditional immunotherapy is centered around this one treatment fits all approach without diving deeper into the patient's unique body and needs. So when we look at this figure here, all of our patients maybe exert the same symptoms at a clinic, and traditional immunotherapy most likely ends here with these patients receiving the same treatment. But their bodies really aren't the same, even if their symptoms are, so each one of them reacts in different ways. Some can react well and others can't. And personalized immunotherapy is a customized treatment option which tailors to the individual needs of each patient. So finding a treatment option this way involves looking at patient and/or tumor genetics and predictive biomarkers and conducting diagnostic tests. So if we bring back our four patients using personalized immunotherapy, each one gets a treatment option specific to their tumor and their melanoma, which suit their bodies better. So here are some immunotherapies that can be personalized, and I'll get to how later in this presentation. So immune checkpoint inhibitors, or ICIs, are drugs that enhance the body's immune system's ability to fight cancer by blocking immune checkpoints, and these prevent overreaction of the immune system. So this figure shows here the T cell, which is this white blood cell, and its receptors binding to the ligands of the tumor cell, and this causes a really suppressed immune response. But immune checkpoint inhibitors are able to release the brakes on the immune system and allow the immune system to attack cancer cells at their full potential. So IPI or ipilimumab is a anti-CTLA-4 inhibitor. And CTLA-4 is a protein receptor. So here we can see that the cancer cell binds to the T cell and activates it. And then CTLA-4 is here at the moment, but it later comes down here and binds to this B7 and kicks CD28 out of the way. So what this does is it inhibits the activity of the T cell and doesn't allow it to work at its full power. So our IPI blocks the CTLA-4 from binding to the cancer cell so that it completely works at its full power. Nivolumab and pembrolizumab are PD-1 inhibitors, so they block PD-1 from joining its ligand, PD-L1, in the same way as the one stated above. So ICIs can be personalized based on certain characteristics of a patient's tumor. So for example, a tumor with a higher mutational burden, which reflects the number of mutations within a tumor, tend to respond better to ICIs and make these patients ideal candidates. And also tumors with higher PD-1 exposure are more likely to benefit from PD-1 inhibitors, and the same concept goes for the CTLA-4 inhibitors. However, these do have their challenges, like ICIs can lead to immune-related adverse events because the immune system is so much more heightened than normal, and not all patients can respond to ICIs due to factors like tumor heterogeneity and individual differences within the immune system. So let's move on to our next immunotherapy, which is oncolytic viruses or OVs. And these are engineered viruses which target and kill cancer cells with the harmful part of them removed. So in this figure, we have our blue healthy cell, and as you can see, the virus does not replicate and do any harm. But in the purple one, it does, and this is called selective replication. And then after that, the tumor cell breaks due to the virus, and this is called tumor cell lysis. So T-VEC is an OV derived from herpes simplex virus 1, or HSV1, and it was modified to remove the gene ICV34.5, which was responsible for nerve issues, but with that removed, it is perfectly safe for use. So in a phase 2 study with advanced melanoma patients, once injected with T-VEC, 8 out of 13 individuals had complete responses, including a significant anti-tumor effect. And CVA21 interacts with ICAM and DAF, which are receptors for viral attachment that are overexpressed on cancer cells. So in another phase 2 study, 38.6% of patients experience IRPFS at 6 months after injection. Personalized OVs are engineered to target genetic alterations specific to individual tumors based on their makeup. The treatment is favorable for patients with a tumor microenvironment characterized by high levels of immune cell infiltration, which is when immune cells are able to migrate from one place to another. And patients that have also failed prior treatments like chemo also use OVs as a salvage treatment option as well. However, if there are pre-existing immunity or rapidly developing neutralizing antibodies, the effect of OVs can be reduced. Also, tumor hypoxia, which is when the tumor lacks sufficient oxygen, can impede viral spread as well. And there are also challenges with off-target effects and systemic toxicity. So lastly, we have adoptive cell therapy, which is where a patient's immune cells are collected, modified, and then reinfused back into the patient to increase efficiency. So here we have our brown cells, which are tumor cells, and they're attacked by various cells like T cells, B cells, NK cells, and dendritic cells. And what adoptive cell therapy will do is advance all these cells that attack the tumor cells. So we have TIL therapy here, which is a type of adoptive cell therapy where the body's tumor-infiltrating lymphocytes, or TILs, fight against cancer cells, as shown in the figure on the previous slide. So there are 5 main steps to employ this procedure. First, a section of the patient's tumor is removed, and then the TILs are isolated. And then these are cultured in a lab to select, some are selected to activate further based on their current activity and efficiency. And then prior to infusion, lymphodepletion therapy is administered, which suppresses the patient's immune system temporarily to create space for the TILs to work properly. And then lastly, the TILs are injected back into the patient with more power and efficacy. Younger patients with lower tumor burdens are more susceptible to this treatment option, and tumors with higher immunogenicity, which is the ability to provoke an immune response and minimal immune suppression, would work as well. However, obtaining a sufficient number of patient tumor samples can be challenging, even if the tumor is small or has low levels of immune infiltration. And there are also chances of adverse events as well, leading to fever and hypotension. So these immunotherapies discussed are all being thoroughly researched and worked on, but another promising direction or a different branch for advancing melanoma treatment lies in the development of personalized neoantigen-loaded nanoparticles. So a neoantigen is a protein that is specific to cancer cells, which signals the immune system that the body is not healthy and that cell is not healthy. And a nanoparticle is a small particle used to precisely deliver drugs. So with this technology, scientists create nanoparticles that carry neoantigens unique to a person's melanoma, and they analyze a person's DNA to find specific markers called peptides from their melanoma and turn these into the personalized vaccine. These vaccines are then transported through nanoparticles to directly attack the cancer cells. And because of this precision, they're able to achieve high specificity, reduce toxicity, and minimize off-target effects. And also, this technology can be combined with other immunotherapies listed previously to make treatment more effective. The end goal here with all these methods listed in this presentation isn't to replace the old, but is to build on what we already have using technology and sciences that are newer to improve therapeutic outcomes and patient survival rates so every patient has the best possible chance at recovery. Thank you for listening. I'll be accepting questions now.
Thank you so much, Shreya. Very interesting. And so many, I feel like we have a really cool combination of cancer-related and astrophysics-related presentations today. So we had a question from Sanvi. So how does CTLA-4 suppression specifically influence the balance between immune activation and also tolerance?
Okay, so CTLA-4 basically stops the immune system from overreacting. So this can cause damage to healthy tissues, right? But if it's suppressed and there's cancer, it'll work on attacking the cancer cells specifically rather than the healthy tissue. So when administered in a balance, this is really able to make sure that one, your body isn't getting attacked in places that it shouldn't be, but it's also focusing on cancer cells itself.
Got it. And we had a question from Samik as well. What is the function of GM-CSF expression as it pertains to replacing ICP 34.5?
So ICP 34.5 is the gene that is responsible for like nerve damage in that HSV1. But when it's removed and replaced with the other nerve, it's completely healthy for use and it targets specifically cancer cells.
Cool. And I actually have one more question, which is how do you, just in general, I'm not a biologist, so pardon me if this is an ignorant question. In general, how do we decide what therapies are better for what types of cancers? Because obviously there are different cancers, right? There are cancers that are solid and an actual tumor. There are others that are sort of like blood cancer, let's say. How is that decided by doctors in terms of what therapy works best for what type of cancer?
So my research primarily was based on melanoma itself, but
Mhm.
doctors primarily use tests like diagnostic tests and other sort of like treatments in the clinic. But the
Mhm.
difference between these standard treatments and these personalized immunotherapies for different cancers is that the personalized immunotherapies work more towards the patient's specific tumor. So in other cancers, they might just like see, oh, they have like melanoma or oh, they have basal cell carcinoma or any type of cancer really, and then employ the standard treatment just based on what's the standard procedure for if they have this cancer. But with personalized immunotherapy, they specifically take that section of the tumor, see, oh, what levels are high in this tumor, what levels are high in that tumor, and make calls based on that.